Hey guys, so this video should be short. I want to say there are like 9 slides in the entire PowerPoint, and that includes cover slide and, the little goodbye slide. So, shouldn't take too long. Now, we've already kind of talked about mutations, but we've talked about them as they refer to an actual chromosome. So now that we've done protein synthesis, and you guys are caught up once more with the method by which we take a gene and create a protein from that gene, let's look at how mutations affect the actual bases, the actual nitrogen bases, the actual sequence that will eventually give us an amino acid, which would eventually give us a polypeptide. So we're going to look at mutations on a molecular scale. So here we go. So let's start off with the different kinds. And we could take them and put them in some major categories. So one of the main kinds of mutations that you often see on the molecular level, and when we say molecular, we're talking about the actual, nitrogen bases that will make up messenger RNA, for example. The first major category are called point mutations. So a point mutation is what happens when a single base is changed. It's also known as a base pair substitution. So let's say we were supposed to have, the base A, and instead we switched it with the base G. That would be an example of a point mutation or a base pair mutation. Now, there are 3 possible outcomes which are also called mutations. The first possible outcome of a point mutation is a silent mutation, where when we make that base change or that base flip, it doesn't actually change the amino acid that that particular codon codes for. Because as you know, the amino acid code is redundant. So we have several codons that code for the exact same amino acid, like lysine, for example. So we may have changed that one letter, but ultimately we are still making the amino acid that we need. So this is called a silent mutation. There is no change in the amino acid, which means that there's not going to be a change to the polypeptide, at least an effective change to the polypeptide, which means that there won't be an effective change when it comes to structure and thereby function. And again, it's the result of the redundancy. Whoops, sorry about that. It's the effect of the redundancy in our amino acid code. The next outcome or type of mutation is called a missense mutation. And that's where, when we do substitute those bases, we do end up changing the amino acid. So we went from lysine to valine, or to histine, or to leucine, or to whatever else. The last kind is called a nonsense mutation. And a nonsense mutation means that our base substitution results in us making a stop codon. So we go from having an actual amino acid to producing a stop codon, which means that wherever this happens, the machinery in charge of translation is going to stop adding amino acids to that polypeptide chain. All right, so to go over, general category is called point mutations. It means that we've changed one base. We've just moved one base for the other. There are 3 kinds. Silent mutations, where there is no change to the amino acid. Missense, where there is a change to the amino acid. And nonsense, where we produce a stop codon. All right, so here's an example. Now, one of the classic ones would be sickle cell anemia. Point mutations lead to sickle cell anemia. So on someone with normal hemoglobin, if we look at them on a molecular level, they would have a codon GAA. GAA will code normally for the amino acid glutamic acid. Excuse me. So this is what we want when we're producing normal hemoglobin. Once we have this GAA and we have our glutamic acid, we're going to get someone that has healthy levels of hemoglobin. Their hemoglobin behaves the way that it should, and that means that their blood cells will be able to carry oxygen the way that they should. That person will not suffer from sickle cell anemia. But on someone who does have sickle cell anemia, look at the difference. Instead of having GAA, we have GUA. So we have had a base substitution. We switched out our A for a U. Well, this changes the identity of the amino acid. Instead of making glutamic acid, now we have valine. So people with Sickle cell anemia have the amino acid valine because of a base substitution. We should have had an A, and instead we ended up with a U. So this would be a missense mutation because we have a change to our codon that results in a change to our amino acid. All right, and we've kind of talked about this already, but I'll kind of just run through it again. One of the main reasons it's such an issue is because glutamic acid is normally hydrophilic, which means that the protein will fold a certain way when glutamic acid is part of the polypeptide, whereas valine is hydrophobic, which means that the protein will fold in a completely different way because valine is hydrophobic. It doesn't like to be around water. We know that this condition primarily affects people of African descent. It is a recessive inheritance pattern, so you have to have both recessive alleles in order for you to exhibit this particular condition, and it strikes about 1 out of every 400 African Americans. All right, so back on with mutations. Another way that you can end up with a molecular mutation is through a frameshift. So you know that we take our piece of messenger RNA and we break it up into sections of 3, or bases of 3 that we call a codon. So those bases are read in what we call a reading frame. So if we were to look at, let me change my pen real quick. If we were to look at this piece of mRNA right here, then we would have 1, 2, 3. That would be our first codon. 1, 2, 3 would give us another. Here's another. And another. So in this piece of messenger RNA, we have 1, 2, 3, 4, 5 codons. And that's because of our reading frame. This is our first group of 3, then our next, and our next, and our next. Well, a frameshift mutation is what happens when our reading frame shifts, where we change which nitrogen bases are part of that particular codon. So it shifts everything, or it moves everything, what we call downstream the DNA. So we push it all down. So a way we could show that, or a way, that frameshift normally happens is by either adding an additional base that wasn't there before, or removing a base that was there initially. So let's look at adding a base first. So when we insert bases, that's called an insertion. We're just putting in one that wasn't there before. So like in this, in this picture right here, this would be an example of an insertion because we are adding in an extra U. So remember, this piece up here is our original DNA. So our original mRNA, sorry. So we're comparing everything to this piece up here. When we add in this extra U, at first, it doesn't look like it changes anything because our first codon is still going to be AUG, just like it was here. But right here, look, we have one codon and another and another. Our second codon, however, is different. Instead of it being AAG, it's now UAA. So we've shifted our reading frame. We've shifted which group of bases are part of the codon, which means that we could inherently change what the amino acid they produces. In the case of adding in this U, UAA codes for stop. So when this piece of messenger RNA gets read through a ribosome, once the machinery gets to this section and that transfer RNA is coming in, it's going to read UAA and it's going to stop. It's not going to add anything more, even though we have bases further downstream. Right? So the opposite would be deletion, where we remove a Here's an example up here. So we'll do like what we did before. We'll count out the codons. So we have one codon and another, and we're moving that U out. So here's our other and another. Well, look at what happens. At first, again, it looks fine. AUG is still AUG. AAG is still AAG. So far, so good. But we should have UUU, and instead we have UUG. But we still have a codon. We're still going to make an amino acid. Granted, it's not the one we need, but we'll make one. If we keep going, instead of having GGC, we will have GCU, We still make an amino acid. It's not the one we wanted, but we make it. But then look at what happens at the end. We don't have a group of 3. So this isn't going to code for anything, which means that our protein is going to be shorter, which means structure is going to affect function. This guy probably won't work. So again, Frameshift, we're just moving which bases are part of the reading frame, are part of the group of three, which makes a codon. And it happens because of insertions and because of deletions. Okay? Now, one thing to keep in mind, if we're adding in or removing 1 or 2 individual bases, it causes a frameshift. When we add and remove 3 nucleotides, which would be an entire codon, we are not really altering the frame. Like if you look at this picture down here, you will see that. We still have, like if we took out that second codon, which is AAG, we will still have AUG. We'll still have UUU. We'll still have GGC. We'll still have UAA. So the the frame hasn't moved, but we will have removed an entire codon, which means we will have removed an entire amino acid. So if we remove 3 codons at a time, or we add 3 codons at a time, we don't change the frame. The frame is still going to be its original group of 3 codons, but we will change the amino acids that go there, or the amino acids that are being removed, or the amino acids that are being placed. Okay, so I hope that's clear. All right, and a classic example of this would be cystic fibrosis, which we've kind of talked about already, but let's refresh our memories. It primarily affects whites of European descent. It strikes 1 in every 2,500 births, 2,500 births. So that means about 1 in every 25 Caucasian people would be carriers. Normally, the allele that codes for our membrane protein that transports chlorine back and forth across the membrane, if that's healthy, if the allele is healthy, then those channels work and we don't have any issues. But we can have defective or absent channels, which will limit how many chlorine molecules we can move and also how much water we can move, which results in mucus buildup that's really sticky and really thick, and then that mucus is going to become an attractive place for bacteria to grow and cause lots and lots of bacterial infections. Without immediate treatment, children who suffer from this particular disease will normally die before the age of 5, if not sooner. With treatment, they can live past their 20s, but life expectancy is still a lot reduced compared to what it should be. All right, so here's an example. This would be healthy. This would be someone with cystic fibrosis that, it hasn't really onset yet. Here we have cystic fibrosis. We can start to see a buildup of mucus. And here, this is what it would look like when the mucus is really built up and there's lots of bacterial colonies growing in there. And this is primarily happening because we can't shift our chlorine, which means that we're going to have osmotic issues. We can't move water freely because of that whole high concentration to low concentration thing. So instead of the mucus being mostly filled with water and fairly movable, it becomes very viscous and very thick and sticky, and it adheres to these linings. All right, and here's the reason. It's because of the loss of 1 entire amino acid. The chromosome that's responsible for cystic fibrosis, or that the disease resides on, would be chromosome 7. And the sequence on that particular section of the chromosome should be what you see here: isoleucine, isoleucine, phenylalanine, glycine, and valine. If a person has all of these amino acids in this order, they normally don't have cystic fibrosis. They don't have any issues, no disease. But if we lose the codons that give us phenylalanine, then we end up with the condition of cystic fibrosis. So this would be an example of a deletion that leads to a major disease. All right, so I hope it wasn't too painful. I hope you get it. If not, you know what to do. Write your questions down. Ask me in class. I will see you guys on Monday and Tuesday. Bye.